Hello guys, welcome back to part 2 of chapter 1. Now, I hope you remember previously we had discussed about the plasma membrane. Now today we are going to discuss about the various kinds of transport system that a cell possesses. As we all see that this part of the cell is the cytosol and the one which is opposite is extracellular part of the cell, and here all the processes are taking place at the level of the membrane, which is separating the cell from the extracellular environment. So number one, we see that we have two kinds of proteins. One is acting as a carrier and another one is acting as a channel. Now the carrier is energy dependent, as we see from this image, where the channel is not energy dependent. The carrier is slower as compared to a channel. Later we will discuss the difference between a carrier and a channel. Another process that is known as endocytosis. Whenever a cell takes in any material, the process is called as endocytosis. Now it can be of two types, either caveoli mediated or it can be receptor mediated. Now caveoli mediated, that is the first one, this one is actually for smaller molecules, whereas the receptor mediated one is for larger molecules. Again, we will discuss about this in details. Just I want to explain this diagram to you. Now, as we see, the caveoli mediated comprises of a protein that is caveolin, which helps in the endocytosis of small molecules, whereas the receptor mediated endocytosis comprises of many steps. First, a coated pit is formed, followed by a coated vesicle. Coated with what? Coated with clathrin, another protein. We will read about that later. Now this coated vesicle forms an early endosome, the pH of which is low, and as it forms the early endosome, it releases the receptor to be used for another cycle of endocytosis. So this receptor can be recycled. Now the early endosome forms the late endosome, and it is this late endosome which combines with the lysosome to form a lysosome late endosome fusion vesicle. So, and this lysosome late endosome fusion vesicle is again reused. So there is a reconstitution which is taking place. So this is all about endocytosis. What is exocytosis? So whenever a cell wants to get rid of some products, it uses the process known as exocytosis. For example, over here, the receptors, the work of the receptor is outside, and when the receptor is recycled, it goes outside by process of exocytosis. Another form of transport is phagocytosis. Actually, it is more of a form of endocytosis, wherein here we have a microbe. A microbe is taken inside the cell and there is formation of what is known as a phagosome. The phagosome combines with a lysosome to form a phagolysosome, and the undigested residual material is again out of the cell. Again, this process is exocytosis. Last process is transcytosis. Transcytosis is a process whereby certain materials are taken inside the cell. No changes take place in that material and it undergoes exocytosis unchanged. Okay, it undergoes exocytosis unchanged. Okay, going to the next slide. Okay. So just wanted to say something about certain materials which are on the outer surface of the cell membrane, known as the glycocalyx, which comprises of the glycoproteins and the glycolipids. Now these are carbohydrate entities present on the external surface of the cell membrane, which has two functions. They act as a chemical and mechanical barrier, and they also participate in cell-to-cell and cell-to-matrix interaction. Now we start with the first kind of diffusion is a passive membrane diffusion. Now we first discuss about the substances which passes and the substances which cannot pass by this method. So let us see what are the substances which can pass by means of passive membrane transport. One is the non-polar substances, for example, the gases like oxygen, carbon dioxide. Second are the hydrophobic molecules, for example, the steroids like estradiol, vitamin D, the hormones. They can cross by passive diffusion. Also, polar molecules can pass, but there is certain limitation. It should be less than 75 Dalton in size. What are those molecules? Water, ethanol, urea. All of these are molecules which can cross by means of passive membrane diffusion. Now, what are the substances which cannot pass? So these are the polar molecules which are more than 75 Dalton. For example, glucose and ions. Keep one thing in mind, that however small the ions are, they cannot pass by means of passive membrane diffusion. So fast, let us revise again. By passive membrane diffusion, what are the three kinds of substance which can pass? They are the non-polar substance, for example, oxygen and carbon dioxide, and the hydrophobic molecules, for example, steroids, and the polar molecules, but should be less than 75 Dalton. Whereas on the other hand, what substances cannot pass? These are the polar molecules more than 75 Dalton and the ions. Now I want to mention at this point certain large channels known as aquaporins which are present in the renal tubular epithelium. It allows the passive transport of large quantities of water present in the kidney. Okay, so these aquaporins are present in the renal tubular epithelium of the kidney, allowing a passive transport of large quantities of water. So how these polar molecules which are more than 75 Dalton pass? So I think, as you remember, we read about the carriers and the channels before. So these are the carriers and these are the channels, as I had mentioned before. So let us see the carrier. What are the differences between carrier and channel? Now carrier is against the gradient, so energy will be required, whereas channels are along the gradient, so energy is not required. Carriers, they undergo certain conformational change and bind a specific solute, so this takes some time, so the process is kind of slow. Whereas the channels, they create a hydrophilic routes for the solutes, and this process is quite rapid in comparison to the carriers. I hope you understood the difference. Now let us come to active transport. So till now we have read about the passive transport, and for molecules greater than 75 Dalton we have carriers and we have channels. Now active transport, energy is always required. Now, what is the main role of active transport? Actually, the main function of the active transport is to prevent the cell from bursting. How come? Let us see. Normally, what happens in a cell, sodium ions continuously leak inside the cell. Now, as the sodium ions leak inside, water molecules also follow sodium ions. So the cell increases in size and swells, eventually leading to cell burst. So if our cells were to function in such a way, all the cells in our body would have swelled up and they had burst. So who is keeping them away from bursting? So for our rescue comes this sodium potassium ATPase. Now what does this do? This pump actually pumps 3 sodium ions outside the cell and 2 potassium ions inside the cell. So normally remember that the concentration of sodium ion is high outside the cell, whereas the concentration of potassium ion is high inside the cell, and there is a loss of one positive charge in the cell because 3 sodium ions go out and 2 potassium ions come inside the cell. Now talking about another proteins which make use of active transport are the MDR protein. MDR stands for multi-drug resistant protein. They pump the polar compounds like the chemotherapeutic drugs outside the cell. So what will happen if cancer cells pump out the chemotherapeutic drugs? The cancer cells will become resistant and will lead to treatment failure. So active transport is also responsible for treatment failure, and the protein in question being MDR protein or multi-drug resistant proteins. Now, one important thing that I wanted to bring to light is that the cytosolic enzyme works at a pH of 7.4, whereas a lysosomal enzyme works at a pH of 5 or less. Now, coming back to another process of cell transport being endocytosis, as I have already discussed in the image, this endocytosis happens in two ways. One is caveoli mediated, which is basically for smaller molecules, and another one is clathrin protein mediated, which is basically for larger molecules. Now, the caveoli mediated endocytosis is also called as potocytosis or cellular sipping, whereas the clathrin protein mediated process is known as pinocytosis or cellular drinking. Now, what are the substances which are transported by caveoli? They are the folate, and this is also used in case of transmembrane signaling. So caveoli mediated endocytosis is used for transport of folate as well as has a role in transmembrane signaling, whereas the clathrin protein mediated process is for transferrin and LDL. So it transfers transferrin as well as LDL. LDL is low density lipid. Going to the next slide. Now, we all remember the clathrin mediated endocytosis. Now, what were the steps? It was receptor mediated and which was followed by coating of the pit formed by the receptor. That was coated by what? By a protein known as clathrin. The pits give rise to complete vesicle called as clathrin coated vesicle, which gave rise to an early endosome having a low pH. Now, at the level of the early endosome, the receptors were recycled out, or they were recycled where? On the outer surface of the cell at the level of cell membrane by a process called as exocytosis. The early endosome giving rise to a late endosome, which finally gives rise to lysosome late endosome fusion. Now, this again has reconstitution. That means the lysosome digests the material which was taken inside the endosome and itself forms a lysosome again, which can be reused for other purpose again. Now, defects. Now, what is very important over here for our NEET entrance exam is this: that in the receptor mediated endocytosis of LDL, that is low density lipoproteins, leads to a condition called as familial hypercholesterolemia. As we all know that the clathrin mediated endocytosis transfers two things. One is the transferrin and another one being your LDL. So if there is any defect in the receptor mediated endocytosis of LDL, that person is going to suffer from familial hypercholesterolemia. With this, I complete the different types of transport in the cell. Now we come to another boring yet very important part of the cell, that is the cytoskeleton. Actually, let us define it. So, ability of the cells to adopt a particular shape, to maintain the polarity, and to organize the relationship of the intracellular organelles and move about depends on the intracellular scaffolding of proteins, and these proteins are called as the cytoskeleton of the cell. There are three main kinds. One is the actin microfilament, second is the intermediate filament, and third being the microtubules. So please note down, these are very important points, exam based. Now actin microfilaments are 5 to 9 nanometer diameter fibrils. They are usually in the form of G-actin. G-actin form is a monomer, and it is also the most abundant cytoplasmic protein. Now this G-actin combines to form F-actin. This F-actin is actually a double-stranded chain. So MCQs very commonly comes in this section, and it is very important that you know that G-actin is a monomer, whereas the F-actin is a double-stranded chain. So now what is the main function of actin microfilament? The main function is muscle contraction. Usually they are present in abundant in muscle cells, and they help in muscle contraction. The other important function is that in non-muscle cells, they help to maintain the cell shape, and they help in cell movement. Now the intermediate filaments, again a very important MCQ is that the size of the intermediate filaments being 10 nanometer. Now there are various kinds of intermediate filaments in different parts of the body. Now these are lamin A. They are present in all the cells, known as the nuclear lamina. Another is vimentin. These are present in the mesenchymal cells. Another one being desmin. Desmin is present in the muscle cell. Neurofilaments. These are the intermediate filaments present in the axons. Then we are having the glial fibrillary acidic protein, also known as GFAP, the short form, present in the glial cells of the central nervous system. And we have cytokeratins. Now there are many types of cytokeratins, and for simplicity they are divided into two main parts, type 1 and type 2. Important is to remember that type 1 cytokeratins are acidic, whereas the type 2 cytokeratins are basic. Now what are the functions of the intermediate filaments? Now as we read about the lamin actually plays a role in nuclear transcription. Whereas the usual function of intermediate filament, a single best answer is that it provides tensile strength and mechanical strength. Whereas the third important thing is that it is a major structural protein in the hair and skin of human beings. I hope I have made myself clear. Third being the microtubules. Again a very important question comes. It is 25 nanometer in diameter or thickness. It is usually a dimer of two proteins, alpha and beta tubulin. It is having a positive and a negative end. The negative end is embedded in microtubule organizing center called as mTOC or the centrosome, which is present near the nucleus, where it is associated with a paired centriole. Now, before I go on to the function of microtubules, I want to mention what are molecular motors. See, molecular motors are nothing but they are proteins that use energy in the form of ATP to move the vesicles, organelles or molecules. See, inside the cell we need some kind of traffic to move some things from one place to another, and for that we are having what is known as molecular motors. Now, how the microtubules function? See, microtubules serve as a connecting cable for molecular motors. Okay, which are the molecular motors in our cell? Kinesins. And which is another molecular motor? We have dyneins, right? So the microtubules are acting as a connecting cable for molecular motors. Okay, so the molecular motors being kinesins and dyneins. Now kinesins help in anterograde transport from negative to the positive end, whereas dyneins help in retrograde transport from positive to negative end. So microtubules, number one, is acting as a connecting cable. Number two function, they help in sister chromatid separation. And number three function, they are actually functioning as a cilia in the bronchi, and they are specialized to form flagella in case of a sperm. So three important function of microtubules. Now we come to cell interactions. Now there are various kinds of cell interactions in our body, right? Number one, they are the tight junctions, also called as occluding junctions. Number two, they are the desmosomes, called as the anchoring junction. And number three, they are the gap junctions. So now we start with the tight junctions. What are tight junctions? Tight junctions are junctions that seals the adjacent cell together and restricts the paracellular movement of ions. Paracellular movements means the movement in between adjacent cells and other molecules. They are composed of three transmembrane protein. Now these are important for MCQ based question. These are, so tight junctions are formed of which transmembrane protein? Number one being occludin, number two being claudin, number three being zonulin, and number four being catenin. Now, as we can see in this image, we can see the tight junctions. Can you see the tight junctions? Yes, we can see the tight junctions. Now, these tight junctions, as you can see over here, we can see the tight junctions over here. These tight junctions do not allow any kind of movement or any kind of paracellular movement because they're adjacent cells between each other. We have also read about the actin filament, as we can see in this image, the actin microfilament, and we have read about the function of actin microfilaments as well. And here, as we can see, as you can see over here, multiple thread-like structures from these are actually the intermediate filaments. As you see, I have already labeled the intermediate filaments, and there are various kinds of intermediate filaments in different kinds of body tissues. Now, coming to desmosome. Now, what does desmosome do? They are attaching the cells, means two different cells are being attached by a desmosome, and they're also attaching cells to extracellular matrix. So they have two functions, attaching cells and their intracellular skeletons to other cells or even the extracellular matrix. Now, there are three types of desmosomes. Again, a very important exam MCQ. One being the spot desmosome or macula adherens. Second being the belt desmosome, and third being the hemidesmosome. Now, before I go into the functionality of the desmosomes, I want to mention few points about what cadherins are. Okay, cadherins are nothing but they are the transmembrane connector proteins, and each desmosome makes use of such cadherins to carry out their function. So first I start with the spot desmosome. They are between cells. Okay, so these form connections between two cells and they are very small, like a rivet. And over here, the cadherin that we use are the desmoglines and the desmocolins. So please note this, an important NEET MCQ question. So the cadherins used by spot desmosome or macula adherens are desmoglines and desmocolins. Now these are associated with intracellular intermediate filament, and the main function of these desmosomes is to transmit the mechanical forces. Coming to the belt desmosome, they are also between the cells, but the difference from the spot desmosome is that they are like broad bands, and here the cadherin that they are using is known as the E-cadherin. They are also associated with intracellular acting, and the main function of belt desmosome is to maintain cell shape and motility. Third type being the hemidesmosome. What is the basic importance of hemidesmosome? It is attaching one cell to the extracellular matrix or the basement membrane, and here the cadherin is the integrin. It is attaching to the intracellular intermediate filament and link the cytoskeleton to the extracellular matrix. Let us go back to the diagram and appreciate the desmosomes. As we can see, the desmosome over here forming an attachment between the two cells. Now here is the belt desmosome, which is forming a broad band connection, and the important one is the hemidesmosome. Why? Because it is attaching not only one cell to another, but this cell to the neighboring basement membrane or to the extracellular matrix, and we can see that the cadherin which is helping the hemidesmosome is nothing but the integrins, and again an important MCQ. Now we are just left with one more, that is the gap junction. Just let me highlight the gap junction. And let us go to the gap junction. Now these gap junctions are 1.5 to 2 nanometer pores. So these are 1.5 to 2 nanometer pores, and they are made up of connexons, which in turn is made up of smaller unit that is known as connexins. What do? They allow the ions, amino acids, sugars and nucleotides to flow through them. They are important in cell-to-cell communication, where mainly in the cardiac myocytes. Again, a very important question for NEET. And they do what? They allow chemical or electrical signals to transfer from one cell to another. It allows the myocardium to behave as what? As a functional syncytium. It allows the myocardium to behave like a functional syncytium. That means the entire myocardium can act as one big unit. So it is allowing the coordinated contraction of the heart. Allows for coordinated contraction of the heart. One thing I have missed in the previous slide are the focal adhesion complex. Again, important for MCQ based. Now they are made up of 100 proteins, and they are localized at the hemidesmosome, and they have proteins that generate intracellular signals in response to stress. Now coming to endoplasmic reticulum or the Golgi apparatus. Now you will be surprised to know how efficiently our cell can work and how sophisticated our cell how sophisticated machinery our cell possess. So first we are starting with the endoplasmic reticulum. Let us see, it is actually the site of synthesis of all kind of transmembrane proteins and lipids for plasma membrane and all the cell organelles inside the cell. It usually specializes in synthesis of those materials which are meant for purposes of export, Again, depending on the presence of ribosomes, endoplasmic reticulum can be divided into rough endoplasmic reticulum and smooth endoplasmic reticulum. Now this is a very, very, very important slide, and everyone should pay attention at this point. Now there are certain nascent protein, actually the mRNAs. I want to stress that there are mRNAs that are transcribed from DNA. If these mRNAs are having N-terminal signal sequence agar present hai, if it is present, then these mRNA will go to rough endoplasmic reticulum and the protein will be synthesized in the rough endoplasmic reticulum. Now after the protein is synthesized in the rough endoplasmic reticulum, few proteins will go and get incorporated inside the endoplasmic reticulum membrane, whereas rest of them will remain in the endoplasmic reticulum lumen. Now over there the proteins are folded, okay? The proteins are folded, and this process is known as oligomerization, and all of this process of protein folding is regulated and happens in front of certain molecules called as chaperones. So what is the function of chaperones? If the proteins are misfolded or they are not properly oligomerized, there are two things that can happen. Either the protein can straight away be degraded, or the protein is retained in the endoplasmic reticulum, and so the endoplasmic reticulum will now try to degrade the protein by itself. Right. Now if this will exceed the capacity of the endoplasmic reticulum to degrade, there will be something called as endoplasmic reticulum stress response, also known as UPR response, that is unfolded protein response, which leads ultimately to cell death or apoptosis. Such an important point. So this concept is really very important, because in subsequent lectures we will go back, because many such diseases will be caused if this mechanism is not done properly, and there is accumulation of misfolded proteins in our body. So in the chapter of central nervous system, there are many such diseases which are caused by this misfolding, one of them being the Prion's disease. Now coming back to the top, if the N-terminal sequence is not present in the mRNA, protein synthesis still will occur, but where? It will occur in the free ribosome, okay, also called as polyribosome, and where? In the cytoplasm. So agar N-terminal sequence is present, the synthesis will take place in the rough endoplasmic reticulum. If it is not, then the protein synthesis cytoplasm mein hoga in the free ribosome. Now, one basic example of a misfolded protein, if this process is not working properly, is this. A common example of a misfolded protein is the CFTR protein. It is because of mutation of a single amino acid at PHE 508 position, which leads to degradation or misfolding or retention of CFTR protein, and the disease it results is cystic fibrosis. Now, we have studied about the role of the endoplasmic reticulum. Let us see. Now, the endoplasmic reticulum ka function kya hai? Synthesis of proteins and lipids for any kind of purpose. Now, whatever is formed in the endoplasmic reticulum now goes, or first it goes to the Golgi apparatus. So, before the product of endoplasmic reticulum enters the cytoplasm, it will make its first go into the Golgi apparatus. Here, the Golgi apparatus में क्या होता है? The proteins are modified as it moves from cis Golgi apparatus to trans Golgi apparatus. So, what happens in the cis Golgi apparatus? If the protein is not up to mark, they may send the protein back to endoplasmic reticulum. But if the proteins are all right, they are normal, they will either go to other organelles or they will straight away be exported to other cells. Okay, so I say again, so from the endoplasmic reticulum, whatever is synthesized, whether it be proteins or whether it be lipids, all of them first will go to the Golgi apparatus. From the Golgi apparatus, the proteins are modified as it will go through the cis Golgi apparatus to trans Golgi apparatus. Now, if it goes to cis Golgi apparatus and if they find that any problem is there, it may send the protein back to endoplasmic reticulum. From the cis, it goes to trans Golgi apparatus, and from here, the proteins will be transported to other cell organelles or it will be exported out of the cell. Now, one important thing I wanted to about the Golgi apparatus is the fact that Golgi apparatus is normally present in those places where any kind of secretion is required. For example, goblet cells in our intestine. For example, bronchus, जहाँ पर बहुत सारा mucus secretion होता है, and in the plasma cell where there is a secretion of large amounts of antibodies. Now, let us say something about the smooth endoplasmic reticulum. I have already mentioned about the smooth endoplasmic reticulum. The important thing is that it is present in increased concentration in the adrenals and the gonads and in the liver. So the increased concentration in the adrenals and gonads is for hormone synthesis, whereas in the liver it is mainly playing a role in lipid catabolism. Again, increased calcium sequestration of endoplasmic reticulum, that is in the smooth endoplasmic reticulum. Again, this is used for extracellular signaling. And third, this smooth endoplasmic reticulum is acting as a sarcoplasmic reticulum in the muscle cells. Now, very interesting part that I am going to teach right now is the protein degradation. What I want to say is that the proteins that are formed in our body, all of them has a life. Okay, and when the life cycle is complete, they need to be degraded in the body. Okay, so there are two kinds of degradation. One is your lysosomal degradation, and second, as in this slide, we are having proteasomal degradation. So let us understand the lysosomal degradation. See, all the proteins, okay, which are senescent, जैसे कोई organ or some organelle inside the cell has become senescent, it gives off some proteins. Okay, any kind of denatured protein, maybe because of UV rays or any other factor, okay, which has got denatured. Okay, there is one more factor which is not shown in this diagram, is that whenever the cell doesn't have nutrition or doesn't have any nutrients, so cell undergoes a process called as autophagy. That means it forms an autophagosome wherein it ingests all the denatured protein or the senescent organ or the organs that are not required okay, by a nutrient depleted cell. And this autophagosome is characterized by the presence of LC3 on the outer surface. This autophagosome will combine with a lysosome, and the contents will, this content that I am drawing is released inside the cell. Either these contents are used up by other cells. Okay, these contents are used up by other organelles inside the cell. So this is one pathway of lysosomal degradation, that is autophagy. Another pathway is heterophagy. Again, it is very simple. Either by the process of endocytosis or by the process of phagocytosis, materials are ingested. In case of endocytosis, can be any material. For example, LDL protein ingested. In case of phagocytosis, the material ingested is a microbe. So both of them will form endolysosome or phagolysosome respectively, and the endolysosome has the same fate as autophagolysosome, that is, the contents are either digested inside the lysosome or they are broken down and the contents are released in the cytoplasm to be used by other organelles in the cell. But the phagolysosome also can end up in the same way, that is, releasing the contents or the contents being digested. But one more thing the phagolysosome can undergo is the process of exocytosis. It can also throw the contents out of the cell by means of exocytosis. Now this is lysosomal degradation followed by proteasomal. Now what is proteasomal degradation? Let us see. Now there are two sides. One is the cytosol, another side being the endoplasmic reticulum. So first of all, a nascent peptide chains are formed. As we know that chaperones regulate protein folding. Proteins are folded, now everything is fine, but because of age, UV, heat, reactive oxygen species, all of these products, the proteins will become what? Old, that is, they become senescent and denatured. By the help of ligases E1, E2, E3, these proteins are attached or marked by a substance known as ubiquitins, and these ubiquitins are recognized by a grinder. If you remember, in the first part I have mentioned about proteasome, they are acting as a protein chewer. They chew the protein, they grind the protein. So these proteasomes recognize the ubiquitin attached proteins and they cut them into small peptide fragments. This was the pathway in the cytosol. Another pathway taking place in the endoplasmic reticulum. Maybe there are many kind of metabolic alterations or genetic mutations or viral infection, means this is not a normal process. Some type of injury is present. So inside the endoplasmic reticulum, there is excess amount of misfolded protein, and we have already mentioned that whenever it will exceed the capacity of the endoplasmic reticulum to degrade the protein, it will go into endoplasmic reticulum stress response, also called as unfolded protein response, which leads to cell death or apoptosis. I hope this is clear. Coming to my next slide. The lysosomes or the proteasomes, again they are having 40 different kind of acid hydrolases. What are the examples? They are proteases, nucleases, lipases, glycosidases, phosphatases, sulfatases. Now we have seen that these acid hydrolases are synthesized in the endoplasmic reticulum, and again anything which is formed in the endoplasmic reticulum will go where? Into the Golgi apparatus. From Golgi they go means they will first go to cis Golgi apparatus, then trans Golgi apparatus, finally they go to lysosomes. In the Golgi apparatus, they are tagged with mannose-6-phosphate. Now, lysosomal degradation, as we have seen, there are two parts. One is autophagy, one is heterophagy. Now, I have already mentioned that maybe because of any intracellular infection or senescent organs or denatured protein, or maybe because of nutrition depletion, first an autophagosome will be formed, which has an LC3 protein attached, followed by formation of autophagolysosome, and then the contents being either catabolized or they are released and used up inside the cell. Now heterophagy, again, which is a part of lysosomal degradation. Now I have mentioned either endocytosis or either by phagocytosis. Finally they form an endosome, endolysosome, or a phagolysosome. Both phagolysosome and endolysosome, the contents are released or they are used up in the cytosol, or the contents will be digested. But the phagolysosome has an extra end. That means it can also go for exocytosis. And going towards the proteasomal degradation, the nascent peptide chain, it depends where it is taking place, either in the cytosol or in the endoplasmic reticulum. So for normal proteins, okay, for normal proteins which have been released and which have served their purpose in life, after they become senescent, after they become denatured because of UV rays or because of aging, they are attached with ubiquitins with the help of ligases, as already shown in the diagram, which are recognized by the proteasomes, and finally peptide fragments are formed. Whereas in the endoplasmic reticulum, maybe because of metabolic alteration or any kind of genetic mutation or viral infection, that is before the protein is released into the cell, excess amount of misfolded protein accumulates, which will exceed the capacity of the endoplasmic reticulum to degrade the protein, leading to ER stress or UPR response, finally leading to cell apoptosis. Thank you so much guys for listening to this lecture of mine. So with this we complete part 2 of chapter 1, and stay tuned and subscribe and like my video, and stay tuned for part 3. I'm sure you're going to enjoy it. Thank you so much for being patient with me. God bless you.